Welcome, everyone. Hope you are enjoying a little bit of spring at this time of year. Welcome to our 2025 ongoing NOAA Fisheries EBFM seminar series. We've started this series with the effort to increase awareness about ecosystem-based management and highlight the progress that not only NOAA are undertaking, but what our partners are doing as well. The library, as you can see, has been an active partner in this effort since the beginning of time, and we offer these seminars on the second Wednesday of each month at this time, 3 Eastern Time. And as Cassandra pointed out, you can access any past presentations in the archive system that NOAA Library maintains. We have a great speaker today, Ryan Vandermeulen, who joined us, I guess over a year and a half ago at the Office of Science and Technology, and has been focused in on formerly working with NASA and focused in on what some of the satellite assets that we have here can do for fisheries management. So it's a very different direction we're taking today. So I want to thank Ryan for joining us. But before I do that and turn it over to him, I want to just announce that we'll have Jenny Bigman joining us on May 14th for our next speaker. Jenny is with our Office of Science and Technology. She's a quantitative ecologist. And we'll be looking at bridging the theory of fisheries science to understanding how fish will fare in a changing climate. So best of luck, Ryan. I look forward to your presentation, and we'll turn it over to you right now. Thanks so much.
Thank you all very much for tuning in today. And Peg, thank you as well for inviting me to speak with this group. So my name is Ryan Vandermeulen. I work for NOAA Fisheries at the Office of Science and Technology. And I'm currently serving as the satellite remote sensing coordinator. It kind of works out that a lot of times, I is spent communicating fisheries needs and requirements to a remote sensing crowd, and it's not as often that I get the chance to talk to a more fisheries-oriented crowd about the latest and greatest in the world of satellite data and technologies. So I'm really excited to have a chat about arguably the highest hill that I will die on, which is integrating more information about ocean color into the fisheries management framework. So I'll start kind of a high level and get more detailed, but broadly, embedded with any given ecosystem are the impacts from climate variability as well as your run-of-the-mill environmental variability. And in the context of fisheries management, one of our central aims is to better understand the full continuum of various trophic level interactions in response to this variability. So as illustrated in the image on the right, there's several components to this. At the very origin of the life forms in the system, phytoplankton growth and phenology are really most directly influenced by the oceanographic environment. And this sets an upper limit on the amount and quality of energy essentially being input to the system. Phytoplankton community composition or dominant size class is also an important factor being influenced by environmental variability. And this can impact how efficiently this energy is transferred through trophic levels. I might be preaching to the choir here, as many of our utilized ecosystem models do take phytoplankton into account in one form or another. But I do want to note that while our U.S.-based fisheries surveys are routinely capturing mid to higher trophic level data, parameters from the lower trophic level are typically not collected, and we often kind of rely on satellite measurements of ocean color to fill that gap. Now, using satellite data has thus far informed a wide spectrum of applications across fisheries. It's been integrated into some of our research track stock assessments. It goes into essential fish habitat models, dynamic ocean management tools, estimates of recruitment and linkages to species richness and so forth. So suffice to say, fisheries is certainly no stranger to the use of ocean color, but there are more layers to this story, especially with regard to new technological advancements. Before I get into that, I want to make sure that we're all oriented. So let's take a moment to talk about some of the fundamentals of what it is we're talking about when I say ocean color, because it's quite broad. So superimposed on this globe is a mapped time series representing the concentration of the photosynthetic pigment chlorophyll A. Now, all life on this planet depends on this molecule, and it's shared by all phytoplankton and cyanobacteria on this planet. So satellite remote sensing of ocean color like this has been really revolutionary in providing an unmatched insight into information about algal biomass as well as biophysical interactions. And this really is our only global insights into trophic energy dynamics that we can observe at this scale, at this sort of global, very repeatable scale. You can see patterns of variability here, even in the most oligotrophic regions on the planet. And having the synoptic vantage point has really lent tremendous value to help understand, hotspots of where marine species live, eat, migrate, as well as make predictions about the future productivity of fisheries in response to environmental change. Getting into the nuts and bolts of this, we're not actually measuring chlorophyll A from space. We're fundamentally measuring essentially the visible portion of the sun's electromagnetic spectrum. So as the basic colors of the rainbow are hitting the surface of the ocean and they go into the water, only one of two things can happen to that light, as I represent with this friendly little photon here. So the light can either be scattered or it can be absorbed by various constituents in the seawater. And once light enters the water column, if we separate those various wavelengths or colors, we see that the quality of light changes with depth as well. So pure water with nothing in it is naturally absorbing more red light, and this allows more blue photons to be scattered back to our eyes, making the open ocean appear blue. Meanwhile, the pigment chlorophyll A is preferentially absorbing red and blue light, actually to fuel photosynthesis. So this leaves green light essentially unused and thus imparts a green hue to productive waters. So some of these photons are lucky enough to be redirected out of the sea surface, back out into the atmosphere, and then into the view of a satellite. And a satellite sensor in orbit is simply measuring the intensity of various wavelengths of light being reflected back. So fundamentally, ocean color science is really just the mathematical, scientific representation of what your eye is telling you, just a little more specifically. So here we're looking at a satellite's view of the ocean after I've mathematically removed some of the atmospheric fuzz to get a little more clear picture. So this particular scene was caught off the Mississippi River Delta, so it's really dynamic. We have a lot of materials coming out of the river here that we see floating in the water, mixing with swirls of algae further offshore, and then these really clear blue waters as we get away from land. So these 5 panels to the right represent the corresponding intensity of visible light being reflected back to the satellite. And you can see that the shape of this intensity or its spectrum changes across these water types. In green waters, the peak of the intensity is over the green portion of the spectrum and so forth. Now, if we look at ocean water anywhere on Earth, we are looking at a complex mixture of optically distinct dissolved materials and microscopic particles that are responsible for changing the spectral reflectance signature of the water, some of which are living, some which are not. Not everything has a color associated with it. Things like salt or dissolved gases or nutrients don't directly change the color of the water, but they may represent processes that impact changes to ocean color. Other things like chromophoric dissolved organic matter, sediments, detritus, phytoplankton, and even macroalgae like sargassum do directly change the color of the water. And if we can definitively link this optical fingerprint to the contents of the ocean, usually by going out to sea and quantifying, physically quantifying the contents of the water, we could then search for this same fingerprint anywhere in the global ocean and infer that the contents of the water share some similarities. And how well we could separate out this information from satellites is largely reliant on how close we can zoom in to see the materials, as well as the range and specificity of colors we're detecting, and the some degree the sensitivity of the satellite instrument as well. So for the last 27 years, since the launch of the SeaWips mission back in 1997, we've only been able to retrieve information in about 5 to 10 discrete bands. This has been enough to derive information about things like chlorophyll A. But this changed in February 2024 when NASA launched its first hyperspectral ocean color mission called PACE. So PACE stands for the Plankton Aerosol Cloud and Ocean Ecosystem Mission. And its big contribution to science here is that we can finally sort of fill in all these color gaps that we've had and really get an uninterrupted sampling across the rainbow and even beyond into the ultraviolet and near infrared parts of the electromagnetic spectrum as well. And from this, we can begin to decipher more specific information about the ocean and things we haven't really been able to see or see very well over the last 27 years. So just to illustrate this, another way, the plot on the left here shows the color resolution we're used to, which we get that one value for red, one value for green, et cetera. And the plot to the right shows the spectral sensitivity of PACE. You can really get a sense of how much extra information we're obtaining here. So there's information here, but what additional insights can we actually get from this? Because there's a lot more information contained within this radiometric signal than simply high or low biomass. Two areas might have the same biomass but represent vastly different ecosystems. And there are over 10,000 species and taxa of phytoplankton, spanning 5 orders of magnitude in size and containing unique assemblages of light-absorbing pigments. Now, while all phytoplankton contain chlorophyll A, additional pigments might be present, including other chlorophylls, chlorophyll B, chlorophyll C, or various carotenoids or phycobilid proteins. And the presence or absence of certain pigments can be indicative of a particular phytoplankton class or species. So these subtle spectral features can contain information that helps distinguish some phytoplankton from one another, like dinoflagellates from diatoms, as well as help differentiate living from non-living materials as well. Now, how cells are collectively scattering light can also inform relative abundance of different size classes. I really like this graphic here from Zoe Finkel's 2010 publication, illustrating that if you scale the smallest phytoplankton to the size of a fish, the large end of the spectrum would be roughly equivalent to the size of Manhattan. And this has obvious implications for what is able to consume these tasty creatures and how efficiently they're able to do so. And just like plants on land, different phytoplankton occupy sort of different ecological niches and support different communities of consumers as well. Some things like diatoms are more likely to be found in recently upwelled waters, really rich in nutrients, and give heavy export ratios. And they're often succeeded in time by more mixotrophic dinoflagellates, which can take advantage of some of the depleted nutrient conditions and even compete with the heterotrophic community. Now, other things like cyanobacteria are very efficient at supporting a more closed microbial loop in oligotrophic environments. And then you have some really out there critters like trichodesmium, which can actually introduce nitrogen into the food web through nitrogen fixation. And there's some cases like in the Arabian Sea, very large dinoflagellates can cause some pretty significant trophic disruptions when it turns out that the only thing that can eat them are gelatinous zooplankton when they have these massive blooms. And in some cases, the phytoplankton are not tasty to anything, and the specific species can produce toxins causing fish kills or getting into cultured shellfish stocks and even killing off large marine animals, which is happening off the California coast right now with I don't know if it's an Alexandrium or Pseudonychia bloom that's happening out there. Now, moving beyond phytoplankton, hot off the press last month, there was this really cool publication coming out of Bigelow Labs. So they were not only able to spot a copepod aggregation from space, they were able to do a pretty bang-up job of estimating their density as well. And they did this by combining and manipulating 3 different color bands of information. The PACE mission offers pretty exciting prospect of enabling up to, I think I calculated 22,100 possible unique sort of false RGB combinations that could be used for teasing out really subtle signals of interest that we can't physically see with our own eyes. Even in the instances that we can't directly sense zooplankton, phytoplankton give off other hints that can help inform potential distributions of zooplankton. For instance, under certain nutrient or light conditions that cause some physiological stress, phytoplankton redirects some of the sun's energy to essentially prevent damage to their photosynthetic machinery, and they actually glow a faint shade of red as they expel this energy as fluorescence. Informally, I like to think of this as cells vomiting excess light as they're being force-fed more food than sunlight than they can utilize, right? But this information can help us consider the progression of a bloom, whether it's growing or dying, does have implications, such as its relative alignment with larval hatching, or the likelihood of finding larger abundances of zooplankton in a fresh versus a decaying bloom, taking into account some of the temporal disconnect between the growth processes. Now, there's also more to the story of ocean color than food and trophic interactions as well. Fish rely on visual cues for basic navigation of space, as well as habitat selection, schooling, shoaling, foraging, prey capture, predator detection, invasion. But there are two aspects to the underwater photic environment to consider. The first is the total intensity of light at a given depth. And we know that light is attenuated exponentially as a function of depth, getting darker. The other is the chromaticity of the water. And depending on what is in the water, different colors are absorbed at different rates. So in the open ocean, blue light is reaching the deepest, while in coastal waters, a lot of the blue light is being absorbed by dissolved organic matter and other things, leaving green light to propagate the deepest. I'll spare you a whole spiel on radiative transfer theory, but one really cool thing is that even though satellites generally measure the surface of the ocean, what we can derive about surface optical properties from ocean color can be used to mathematically propagate light into the water column, and thus be able to make an estimate of light availability and its color and chromaticity at depth. And this is exactly what Chris Hake from the Northeast Fisheries Science Center did. So Chris paired satellite information about light availability and light quality at depth, and he paired this with years of fish density data from our trawl surveys. And he found out that many species are more likely to be found under certain subsurface light conditions. So the plots on the left here are displaying the locations of the Northeast trawl survey, the sample stations there. And then the color scale shows two parameters that were derived from the satellite optical data superimposed on there. So on the left, we see a descriptor of the chromatic environment at the seabed. So this has been propagated to the seafloor. Essentially, bluer colors here correspond to more blue waters. And as we approach the shoreline, the bottom waters become more green, yellow shifted. On the right of those two, the total amount of incident light was also propagated to the seabed. And what you're seeing here is essentially the light intensity at the seafloor. Now, moving your attention to the contour plot you see on the right, this is the output of a species distribution model that was trained with this optics data. And what it's showing is the estimated likelihood of finding, in this case, summer flounder as a function of light intensity on the vertical axis, and then the color of the water on the horizontal axis. So the way to interpret this is basically the brightest colors are showing the optical conditions where the species is most likely to be found. The further down, the darker it is, and the further to the right, the more sort of green yellow shifted it is. So here this is just indicating a species preference for more green shifted waters, but under a relatively wide range of ambient light levels. And I think this was for spring specifically. The question becomes, is this simply a more fancy covariate of depth, or are we actually seeing some, capturing some responsivity to the light environment? So I want to show kind of a cool case study here that Chris did. All right, so here I'm showing the estimated species density for two species of dogfish, and this is based again off of the satellite optics data. And walking through the graphical interpretation here, so these plots predict that smooth dogfish are more likely to be found in darker, more blue shifted waters relative to the spiny dogfish, which tends to inhabit brighter and slightly more green shifted waters. What was really cool is that Chris was able to independently kind of corroborate this optical niche hypothesis. And one means of doing this is through electroretinography. Kalinowski et al. In 2014 gave these same two shark species some basically a form of anesthesia, and while asleep, they measure both the luminous and the chromatic sensitivity of the eyes. And they also measure something called the flicker fusion frequency. You could think of this more as it's like the integration time of the eye. If you're a species that occupies a darker environment, your eyes will spend more time collecting light before sending a signal to the brain, essentially to help build up enough signal. If you bring that same species to the surface, their eyes could get very saturated as they're kind of flooded with too much light. So if you're a species occupying a brighter environment, you take sort of shorter samples at a higher frequency, and at a higher frequency, you're sending these signals to the brain sort of more quickly, right? So in this study, they found that the smooth dogfish exhibited this slower integration time relative to the spiny, meaning physiologically they can adapt better to dark waters, which is also where we happen to find them based off of the survey and satellite data. There are also inherent differences as to how these two species sense light. To the right are some plots taken from the Kalinowski paper. Now, on the bottom right plot, we see here that the spiny dogfish shows a broader spectral sensitivity from 500 to 600 nanometers here, as well as this really cool adaptive kind of diurnal response to more green shifted chromatic sensitivity during daylight hours. This suggests that spiny dogfish are better adapted to brighter environments relative to the smooth dogfish, because if you are deeper and darker, remember it's not just the total amount of light, but the quality of light. There's not as many colors of the rainbow that are actually reaching depth, so you don't need as much of that broad sensitivity across the visible spectrum. They also found that the spinies had a much quicker integration time, which also supports their ability to occupy brighter waters relative to the smooth dogfish. And it's in these brighter, more broadly colored waters that we tend to preferentially find the spinies. Anyway, I really thought this was pretty neat, and that you could kind of see these physiological differences kind of corroborated by independent estimates of model distribution likelihoods based on sort of water optical niches. Taking this one step further, Chris also put forward a hypothesis that these light parameters would offer more predictive capability in species distribution modeling than a static parameter like depth, which is very commonly used. I don't want to belabor the point, but in assessing the performance of a species distribution model trained with these satellite data parameters on light quality, he found that besides temperature parameters, the optical niche was the most influential predictor, explaining on average about 16% of the variance across all 40 species that were in this analysis. Now, light quality, likely co-varies as a term, with depth as well to some degree. But Chris found that models which incorporated the optics in place of depth outperformed the models using depth alone as a predictor. So the advantage of using light here is that it's a more dynamic variable that has the ability to change in response to environmental or climate variability. Whereas if you have large temperature shift, your depth kind of remains static, right? Anyways, I'm working now with Chris to use new data from the PACE satellite to improve on this, and then transition a tool to the Northeast Fisheries Management Council, members of which who are actual collaborators on a proposal as well. So at this point, With this, I hope to have convinced you that there's a lot more going on in that color signal than just the chlorophyll, right? And also this information has relevance to fisheries management. But don't leave yet. There's still more to this story that impacts NOAA fisheries. So colloquially, when we're talking about ocean color satellites, we're referring to measurements that are collected by polar orbiting satellites, which, as you can see here, means it's able to kind of circle around the globe along a vertical axis, and it's measuring from pole to pole, eventually giving us a global snapshot of the Earth. Now, how much of the Earth is covered in a single day is largely a function of how far we zoom in. In general, the more detail you have to zoom in closer and you have a smaller swath, and it takes a longer period of time to sort of get a global picture. Now, alternatively, we have what is known as a geostationary orbit. This is the type of orbit that our communication and weather satellites fly at. And they're located about 50 times further out from Earth than our polar orbiting sensors. And thus, they're collecting data a little differently. So here, we're orbiting horizontally along the equator, but the orbit speed is the same speed as the spin of the Earth. So from the satellite's perspective, Earth is essentially standing still. And this allows us to get multiple views of the Earth on a given day. This is how we can create movie loops of hurricanes and storms. Now, since 1975, NOAA has been operating a series of geostationary sensors that have really solely been kind of dedicated to the weather side of the mission. But that is changing. In the coming decade, this weather mission will evolve into a full environmental monitoring suite, looking at air quality, water quality, fire, storms, lightning, floods, volcanoes, really addressing all of NOAA's key mission areas through the geostationary extended observations program, or GeoXO. So this is essentially the follow-on to the GOES-R missions that we get some of this storm data from. Now, included in this suite is an instrument for measuring ocean color, which would represent a new capability for the U.S. in providing this hyperspectral data that PACE can provide, but at a higher resolution and at this sort of cadence that we have yet to accomplish, getting these multiple views. This adds several layers of extra utility for ocean color data products. For one, multiple looks per day helps us fill in the gaps from our very cloudy Earth. And on day one, we'd be getting 2 to 3 times better coverage, just actual geographic coverage, because we're able to kind of mitigate some of this cloud cover by waiting for the clouds to pass throughout the day. And then also sequences of imagery on the ocean allows a lot more, real-time operational capacity. So, where dynamic ocean management is concerned, we can better rely on the availability of color information sort of on demand. And 3, it's going to see a much finer spatial footprint that enables this color information to be useful at the coastal and estuarine scales, not just the open ocean. This is typically what we're looking at with a 1 kilometer spatial footprint that we get from most of our current satellite constellation. So with GeoXO, we'd be getting on the order of about 300 meter spatial resolution, where suddenly we can see dynamic ocean features relevant to scales that, say, a recreational fisherman can travel over the course of days. This sort of scales that are meaningful to humans here in our everyday lives. So everything about this ocean color instrument specification on GeoXO is specifically designed with coastal and nearshore applications in mind, applications concerning human health, food safety, recreational activities, hazard mitigation, environmental impacts, and overall ecosystem health. So just to give you an idea of how this would work, there will be 2 ocean color instruments in orbit at the same time, and they'll be launching in the 2030 time frame. And I know it kind of seems like a long way out, but in satellite time, it takes a long time for things to happen. Most of the planning is happening now. So one of these satellites will be looking at the East Coast, Great Lakes, and the Gulf, Puerto Rico, and the other will be looking at the West Coast, Alaska, and the Pacific Islands. You can see with these red boxes here. They're essentially representing the proposed coverage area of the GeoXO mission that the ocean color instrument will be seeing. Beneath that are the annual tracks of the NOAA white ships for 2022. And first time I saw this, it kind of blew me away. You know, no other government organization spends as much time at sea in these specific areas as NOAA. And within NOAA, a good chunk of that time is devoted to our fisheries trawl surveys and other scientific missions. So if we really want to get the most use out of these advanced satellite capabilities, it stands to reason that we should become really more familiar with interacting with this data, perhaps even so far as to collect this very type of information alongside the parameters that are of interest to fisheries. So we asked, why not put a miniaturized version of one of these satellites on the vessels themselves? So as part of a strategic initiative to help integrate remote sensing capabilities into NOAA Fisheries, we're doing just that. We've bought 2 hyperspectral radiometers, and they attach to the foremast of the ship, and they make continuous underway measurements of ocean color from sun up to sundown. And as we traverse, these different water types, we'll accumulate a unique set of these, optical fingerprints associated with changing levels of dissolved organic matter and detrital particles, phytoplankton types and abundance, suspended sediment, all of which is defining the habitat of the residents of the sea, which can be more, tricky in coastal regions where we're catching 95% of our fish. So just to get a closer look here, the system we settled on is called a dynamic above water radiance and irradiance collector, or for you Doctor Who fans in the crowd, the Dalek. And you can see a bit of the resemblance from the instrument in the upper right-hand corner here. So the sensor is measuring the total amount of incident irradiance coming from the sun while simultaneously measuring the ocean and sky radiance. So with these 3 measurements, we can derive the same exact base unit of ocean color reflectance that we're getting from space. It's also a hyperspectral sensor, just like the PACE instrument and our NOAA's future GeoXO ocean color instrument. So a double plus bonus there. Now, this consists of a whole integrated system. This is super cool. The sensor itself is mounted to a boom pole, and on the end is a gimbal that allows the sensor to kind of keep a constant flat view of the ocean while you're on a rocking ship. And behind the instrument is an integrated GPS unit. And what this is doing is tracking the ship's location and heading, and knowing where it is, where it's going, and the time of day. The sensor automatically rotates along a horizontal axis to avoid sun glint and only look at the optimal view angles and consistent view angles throughout the day for measuring. And this all connects to a portable deck unit, which can stand alone, or it can broadcast data directly out through an Ethernet connection. This is almost completely automated and maintenance free. It's literally a factor of just turning it on and off. I trained a postdoc to operate it in less than 5 minutes, and the data turned out great. So our maiden voyage was just this February on the Henry Bigelow as part of the EcoMon campaign, and also part of a simultaneous PACE validation campaign. I had no idea before that the foremast can come down like this, but the text brought it down, which was really helpful. And I was able to mount this setup to the foremast using a series of U-bolts and brackets and an aluminum plate, essentially. So just when I thought I was kind of maybe getting soft in my headquarters role, I was very grateful to have the opportunity to play with some machines again. That said, I had some sleepless nights. I was incredibly relieved when it came back in the same condition that I left it, and not at the bottom of the ocean. Didn't want to have to explain that. Anyway, so while continuously measuring ocean color underway, rain or shine, cloudy or sunny, we're getting these constant updates of the optical fingerprint of the ocean. The plot to the left is actually a comparison of our radiometer data in blue against 2 overpasses from the PACE satellite that happened on that day. We're getting these spectra like this every 5 minutes. It's being averaged over 5 minutes. And we get a standard deviation from that as well. And at the same time, from this, we're able to derive the entire suite of ocean color products. So to the right is the actual data parameters that were calculated from this light spectrum we collected on the Bigelow. So not only are we getting chlorophyll, but also estimates of particulate and dissolved carbon pools, underwater light conditions, water mass classifications, suspended particles, CDOM, detritus levels, and more. The salinity and temperature and wind speed are coming out of a model data that matches with the time of collection. But we haven't yet, we could also tap into the ship data for this. So it's also spitting out data in a format that's directly compatible for submitting satellite validation information to NASA. So you don't have to stress over getting all like the header information just perfect, which can be a real time suck. So now the aim of this project is not to supply satellite validation. It's a nice perk, and it serves to benefit NESDIS as they assess satellite quality under varying oceanic and atmospheric conditions. But for fisheries, we're in this to amass a large amount of optical and biogeochemical data at the same time and place that we're doing our surveys and conducting other science. Really, the primary aim is to collect and integrate more information about lower trophic levels, as well as the biogeochemical and optical conditions of the habitat. And it makes sense to have this information coincident with data already being collected aboard fisheries surveys and at the same time and space scales. And we want more of it because many of our ecosystem models, specifically our Cepheid models, they're thirsty for biogeochemical validation. It's important that this information is accurate because really fairly innocuous uncertainties embedded in the lower trophic level parameters with numerous dependencies invariably cascade into much larger model output errors, right? And these uncertainties kind of make it more challenging to assess ecosystem impacts from environmental change. Finally, this data set's also intended to really engage NOAA Fisheries in the socialization of these hyperspectral data streams and make it simpler to develop fisheries-specific applications from future NOAA satellite investments like GeoXO. The work for developing those algorithms and those products happens now, during this phase. So we've generated a lot of excitement amongst our scientists already. Although travel's up in the air, we did, we had these sensors booked for the remainder of FY25. OMAO is also aware of these efforts. We've been talking about integrating these radiometers into their ship-to-shore API system that's being installed on all the NOAA vessels. So with that, if we could tap into that, we could actually do real-time processing of the data and not even have to be on the boat to do so. And so, we're looking right now to leverage data collection opportunities, especially where there's other relevant parameters being collected that can be informed or enhanced from having ocean color data. So we have a lot of simultaneous efforts underway right now to kind of additionally socialize these new data capabilities from websites and podcasts to fisheries specific hack weeks and even direct funding of satellite product development coming from fisheries and in cooperation with NESDIS as well. So one thing I'd like to bring attention to is that there's a new tech memo that will be released soon. It's under editorial review, so hopefully within a week or two. It explores sort of all the applications of hyperspectral ocean color data, specifically with regards to how it applies to fisheries and aquaculture management. And it's meant to be very approachable. It kind of covers a gentle introduction to ocean color, different satellites, and help with sort of finding the data and what sensors you want to use. Then it goes through 16 different satellite products, and for each one, it explains, what the product is, how it impacts fisheries or aquaculture, any limitations or caveats, and also explains how hyperspectral data specifically can improve or has improved this product. So do stay tuned. Because it's under editorial review. Hopefully, it'll be clear for release in the coming weeks. And I think I will wrap it up there. I really appreciate your time and attention here, and I'd be delighted to answer any questions you have or any feedback on the project. So, thank you.
Awesome. Thank you for your presentation, Ryan. I do think we have had at least one question come in so far. So let me pull that up for us. How do those Dalek sensors differ from airborne hyperspectral sensors, like those that operate on LiDAR flights, etc., in coastal zones?
Yeah, it's a good question. In a lot of ways, they can be very similar. They have similar specifications, measuring the same wavelengths of light. They do measure a little differently. On a flight, you're at the mercy of the direction of the plane and making um, um, measurements at a given angle that may be sort of suboptimal. So you might have to filter through a lot of the data, but it's really useful. Obviously, even if you have to filter through data, being on a flight offers a lot more possibility to collect a lot more data in a shorter period of time. So, in short, they are similar, but there's different. They operate differently as well. The shipborne ones are simultaneously measuring both the all the light coming down, but importantly, the skylight at the same angle that the ocean is looking. So, as you're looking at the ocean, you're getting reflected skylight into that signal. So part of that correction, is done with that sky radiance information. It's possible to do from drones, from aircraft. It's just a different process and more complex and can introduce some uncertainties.
Our next question is, you showed a neat example of zooplankton detection for Calanus in Gulf of Maine. Are there examples of zooplankton detection in other regions? How about for gelatinous species that are often hard to sample with nets?
There are, and I'm blanking on the exact locations of that, but I have read some literature that has shown this sort of detection of really large, um, you know, aggregations. What was unique about this recent paper is that they were able to kind of calibrate that signal and be able to estimate a whole range of specific concentrations, and not just sort of a yes or no. There's an aggregation there. Um, what was the second question? Sorry.
Oh, sorry. I guess I should have left it up a little longer. How about for gelatinous species that are hard to sample with nets?
Yeah, yeah. I haven't heard of that. Um, so no, I'm not quite sure. I don't think I've come across it.
Okay, thank you very much. Our next question is, sorry if I missed this, but are observations from Dalek compared against water samples collected on board the ships?
Yeah, that's the idea. This for the Bigelow campaign, we had a whole slew of other optical and biogeochemical measurements on board. The optical parameters are really useful in helping us get to the nuts and bolts of why the signal is the way it is. The specific way, we can measure the specific way that light is being absorbed, scattered, the directionality of that scatter all changes, based off of the unique composition of the water. But I think they were also doing, several, biogeochemical parameters through filtering. But that would be great. That's what we would be calibrating for, essentially, and trying to build up the types of measurements that we make on the ship. And having this radiometry available kind of helps create that link, so we can make some definitive connection between this optical signal even if it's not directly, like I mentioned in the case of fish, like the color signal or chlorophyll, they may not necessarily be responding to phytoplankton availability at any given time, given sort of the spatial the disconnect in size. But they can be responding in real time to sort of the optical clarity and the changes in the environment that are being manifested through this color signal. As you get upwelling or eddies, you can see it's kind of manifesting these physical oceanographic features as well.
Okay, our next question is, can these sensors distinguish different phytoplankton groups, diatoms, dinoflagellates, etc.?
Yeah, it's the $1 million question. There is the first algorithm that's officially been released by NASA is called Moana. I can't remember what it stands for, but it puts out an estimate of Prochlorococcus, Synechococcus, and Pico eukaryotes. So these 3 different classes. This is really geared towards more offshore waters. You're going to find your Prochlorococcus in kind of the most oligotrophic waters, and the Synechococcus, maybe on sort of the boundaries of some of those gyres, and a lot more of the Pico eukaryote concentration starts to pick up closer you get to the coast. That's one that's out. There are plans to release other algorithms that have been demonstrated, but they haven't been approved yet by the PIs who made them. They want to make sure they can demonstrate them before they sort of release it to the public. There's one that's going to distinguish haptophytes, dinoflagellates, diatoms, cyanobacteria, 2 more. And there's another one that's going to look at pigments as well, at HPLC pigments, and try to distinguish that. It's really cool. It's essentially taking you're trying to pare down that signal by comparing it against a model, and kind of taking the residual signal and emphasizing all those bumps and wiggles to where you might find absorption peaks for various pigments. There's some size class stuff that's available right now. There's been available some that are kind of based on empirically based on temperature and chlorophyll that give you estimates of pico, nano, and micro phytoplankton. And then there's some other ones that give you a whole particle size distribution, a continuum of that size distribution. But those have yet to be implemented sort of operationally. If you are interested, that is information that is very helpful to me to help advocate to get these things operational. If I have a use case for them and I have people demanding the data to further the fisheries mission, that holds a lot of weight, and we can push these algorithms through.
Our next question is about pace. Can we get reliable information in optically shallow waters, i.e. 30 meters or shallower?
Yeah, it's been really tricky to get sort of good optical information for some time in coastal waters, both as a factor of you're getting a lot of potentially, especially if you're near an urban area, you get a lot of absorbing aerosols that blow off of the land. They're really hard to correct for. You don't know exactly sort of what they are, and they absorb really a lot into kind of the blue and UV light range. So historically, we've gotten kind of these, you'll see negative reflectance values that pop up in the coast because of these sort of issues. And then you have a problem with the data products too, that are, if you're trying to estimate something like chlorophyll, you're doing it based off the greenness of the water, essentially. But there's other things affecting that greenness of the water. There's CDOM and detrital materials, and it's hard to disentangle those two. With PACE, having this extra hyperspectral information, what was attempting to be done essentially is utilize this extra information, even as it goes out into the ultraviolet, where there's not as much of a phytoplankton signal, but a more dominant exponential detrital and CDOM signal, to find these areas where where one dominates over the other, so we can really hone in on these these products and make sure that we're distinguishing them. There are efforts underway right now from NOAA Fisheries as well as NASA collaborating to make a coastal chlorophyll algorithm specifically for this, so we can kind of get rid of that bias. That's the idea, kind of getting better optical properties and being able to distinguish that better with more of this spectral information.
All right, thank you very much. Our next question. Nice presentation, Ryan. Are you aware of anyone using PACE in predator species distribution models yet? Whales, tunas, sharks, etc.
Not in not PACE yet, just because kind of fresh. It's fresh data from last February. I haven't seen it yet. Doesn't mean people aren't using it, but I do hope to help socialize this and kind of help raise the recognition that these sort of new data parameters are available, which might be more useful than what Ocean Color has offered in the past. It does, using chlorophyll certainly makes its way into several of the predator SDMs, Top Predator Watch from the Southwest, and Whale Watch, and into the Marine Mammal Viewer in the Northeast as well, and some others that incorporate primary productivity or ocean color chlorophyll information.
All right, thank you. And then we did have a question that came in the audience chat. Does the Dalek pick up low concentration blooms better than PACE?
The cool part about having this information on the ship is that the ocean is a really dark target from space. A lot of these, if you're building an ocean color sensor, it has to be optimized for a dark target. It often doesn't work over clouds or land because you need to sort of open up your aperture for long enough to collect that dark signal. 90% of that signal that you're seeing from space is all atmospheric fuzz, and only about 10% is the ocean signal. So there's a lot of uncertainty in trying to remove that atmospheric component, and that's one really helpful aspect of having these Daleks on board, is that you have that sort of unbiased signal that you don't have to worry about the atmospheric component. But it should have similar forms of sensitivity. What's absolutely amazing to me is the sensitivity of PACE in space is such a complex instrument. It is more sensitive and performs optically much better than most of our in situ instruments. It's really a feat of engineering. But yeah, I mean, having just getting rid of that atmospheric signal alone, I think you have a better chance of seeing some of those subtle features. But even so, from space, really, I mean, you can see changes in the middle of the South Pacific Gyre. It's very, very sensitive.
All right, thank you very much. And that is all of the questions that we have had come in so far. I'll give it another few seconds just in case anyone was in the middle of typing a question. And then if nothing comes in, we can wrap things up.
Oh, I just want to take a moment, Ryan, and thank you so much. That was a wonderful presentation. Really opened a lot of folks' eyes and opportunities ahead. So I appreciate all your efforts in sharing these ideas. And for all those folks that would take are interested and couldn't attend, I'd recommend sharing our link to our archive site with your colleagues, and you can see the presentation later on. So thanks to Cassandra and also to Ryan and to all our attendees. I appreciate all the ideas that you offer. And come back on May 14th and meet Jenny Bigman. Thank you.
Thank you, Peg, for organizing today's event. And thank you, Ryan, for sharing your work with us today. Audience, we hope to see you next month. Like Peg said, May 14th. Thank you all for attending today and enjoy the rest of your day.
Thank you. Appreciate it.